I'm going to start with the topic of today, which is LC-MS in biopharmaceutical analysis. And of course, first of all, a warm welcome from me as well. Since my last update dealt with the analysis of biopharmaceuticals in general, today I would like to take a closer look at the role of LC-MS as a technique for the analysis of these drugs. So, of course, 30 minutes is not really enough to exhaustively cover the topic of today's update. After all, others write complete books about this topic. So if you want to delve deep into the subject, I can recommend, for example, this book. But yeah, you really have to be very interested in the topic if you want to go through the whole book. Anyway, I hope that this update today is the basis for an exchange on today's topic. And because of this, you are invited to contact me or my colleagues at HWI to discuss the topic in more detail if you want. Today, I would like to use a few guideline-related examples to explain what we can use LC-MS for in the analysis of biopharmaceuticals. So, to recap, biopharmaceuticals are drugs produced using biotechnology. They are typically derived from living organisms, such as bacteria, yeast, mammalian cells, or plants even. And they include, for example, proteins, of course. Insulin maybe is the oldest and best known example of a biopharmaceutical. But biopharmaceuticals are also enzymes, for example. Alpha-galactosidase A is an enzyme that is used in enzyme replacement therapy in morbus Fabry, for example. Then we have monoclonal antibodies, which are, if not the biggest group, definitely the group with the highest value at the moment. They are used in different indications, for example, in cancer therapies to deplete B cells, or Humira, for example, a very old example, as an inhibitor of TNF-alpha in different inflammatory indications. Then, of course, we have mRNA-based vaccines. So the vaccines of Pfizer, for example, Pfizer-BioNTech and Moderna that were used in the COVID pandemic. And we have all these gene and cell therapies that are more and more coming to market. CAR T-cell therapy, for example, in B-cell lymphoma. So very different kind of therapeutic approaches are collected together under biopharmaceuticals. And sometimes we find also the term biologics interchangeably with biopharmaceuticals. We understand under biologics biopharmaceuticals plus products derived directly from blood, for example, erythrocyte or red blood cell concentrates, erythrocyte concentrates, or other products used in transfusion, and also other kind of vaccines, dead vaccines. In other words, if the information of a nucleic acid is not directly used to produce these products, these therapeutics, then we call them biologics. So biologics is maybe an even broader term that includes biopharmaceuticals and these other products like blood-derived products. But I have to say that this distinction is not strictly adhered to. Anyway, this overview I borrowed from analysis of the Boston Consulting Group. And I don't want to go completely into detail, but what it tells us is which therapeutics are to come, let's say. We have on the right side therapeutics that are already very mature, so they are on the market, longer on the market already. We see all these small molecules, of course, monoclonal antibodies. And then going to the left, the maturity of the approaches decreases, but I think it's fair to say that when we look at it, that the complexity of these therapeutics that we see here and that are advancing and emerging, the complexity increases more and more. So what does it tell us? So these are the therapeutics of tomorrow and tomorrow after tomorrow. Yeah, so this is what is going to come. And because of this increasing complexity, of course, our analytical techniques that we use to analyze these products have to become more and more complex, more and more sensitive, and more and more sophisticated, maybe. So they have to mature, they have to grow together with these kind of therapeutics. And this is true because if we directly compare what we are used to, all these chemical drugs, then they are small and well-defined, whereas biopharmaceuticals are large, they are heterogeneous. Even one biopharmaceutical is often not a homogeneous product. And that's why all the techniques that we use to analyze them they are more, and they are even, if we look, for example, on mass spectrometry, we have to use high resolution mass spectrometry for analyzing biopharmaceuticals. We have other challenges. For example, stability is a challenge when it comes to biopharmaceuticals because they are prone to degradation and aggregation. We have variability from batch to batch. Yeah, so they are not that highly reproducible as chemical drugs, but because of their complexity, they require advanced monitoring during production, of course. And also, all regulatory tests are not standard anymore. So we don't have these standard purity and identity checks, but we have to address things like immunogenicity and biosafety. So biopharmaceuticals definitely need a much more complex, more sophisticated analysis than chemical drugs does. And this is, of course, also true when it comes to LCMS and to our LCMS equipment, of course. And that's why we need systems that have really a high resolution. So let's say these triple quads that we often use to analyze small chemical drugs, they are workhorses. They are sensitive, reproducible, they are perfect for quantification, but they miss the high resolution that we definitely need in these complex biopharmaceuticals. So that's why there are more or less two kinds of analyzers that we use when we do MS on biopharmaceuticals. On the one hand, these are time of flight analyzers, where the mass of the analytes are measured by their flight time through an evacuated tube. And on the other hand, we have Orbitrap analyzers, where the molecules in the analyzer rotate around a spindle. You can see it maybe here, and oscillate from side to side. And the speed of this pendulum motion it depends on the mass of the molecule. Yeah? And to analyze the huge amount of data that this analyzer produces, we need a mathematical technique that is called Fourier transformation. But don't worry, I don't going to talk about Fourier transformation today because yeah, that would crash this virtual update definitely. So HWI uses for the analysis of biopharmaceuticals a cycloxenotroph that comes with some interesting opportunities like electron activated dissociation. So it is very well suited to analyze biopharmaceuticals because of course it has this high resolution that we need. Okay, so if we analyze proteins in LCMS, I'm going to focus today on proteins or protein biopharmaceuticals, then we distinguish more or less 3 different modes that we use. The first one is what we call bottom-up proteomics. So what we do is we digest the proteins of interest with an enzyme. Often trypsin is used with an enzyme that has a high number of recognition sites inside of the protein. So what we receive are quite small peptides. And these peptides are then separated by liquid chromatography. They are transferred to the mass spectrometer, and there we do a peptide analysis. So we can analyze the sequence and the size of the peptides. And then we have another mode that we call middle-down proteomics. Here we use other enzymes. These enzymes, they do not have that many recognition sites inside of the protein, so we get larger peptides. And everything else is more or less the same. But because of these larger peptides, we often have a better resolution when it comes to the analysis of post-translational modification, for example. Especially glycoforms are much better addressed by using these larger peptides than using these very small tryptic peptides. And then we have a third mode where we do not digest at all. So we analyze the intact proteins, and if the protein is very pure, we can directly infuse it into the mass spectrometer. But in most of the cases, we also use LC to separate the proteins. And the advantage of LC, of course, is also that we can easily automate this. So we can handle a lot more proteins or a lot more of products because it is automatically infused into the mass spectrometer. And then we choose from the MS1, we choose the proteoform that we are interested in, and we can analyze this in MS2 mode. So these are more or less the 3 modes that we use, and I will refer later on in the virtual update from time to time to these modes. Okay. If you want to find out which analysis is necessary for your product, you generally follow the relevant guidelines. I have picked one here. It's the ICH topic Q6B guideline that is about the test procedures that are necessary for biotechnological and biological products. Yeah, so of course there are other guidelines like Q5C that tells us something about stability testings of these products, and of course all these EMA and FDA guidelines where you also can have a look at. But I choose this one as an example. And if you look in the table of contents here, then you see that there is an area where specifications are given. I zoom it in. And here you have the specifications, of course, the drug substance specification and the drug product specification. And we see that under the drug substance specifications, we have to look for appearance and description. We have to look for identity, purity and impurities, potency and quantity. And LC-MS is mainly important in these three categories. So identity testing, purity and impurities, and also quantity, because if you do it right, mass spectrometry is also quantitative. Potency, we need bioassays. Sometimes we can check for enzymatic products by LC-MS, but mainly we need bioassays and things like this. So LC-MS is not the main technique for potency. And in appearance and description, we have visual inspections, for example, in this part. Oops, sorry. More or less the same is true for the drug product specification. Yeah, so also identity, purity and impurities and quantity is the field that we address with LC-MS mainly. Okay, so if we want to have a closer look, so we really want to know what we have to test, at least in the ICH guideline here, we can have a look in the appendices. And the appendices then tells us that we, for example, have to test structure, or we have to do a structural characterization and confirmation. What does that mean? Under structural and characterization and confirmation, we find that we have to address the amino acid sequence, we have to address amino acid composition, terminal amino acid sequence, peptide map, disulfide bridges, carbohydrates. You can even make the list longer, but this is what is given in the ICH guideline here. Okay, and of course, amino acid sequence is LC-MS is a perfect technique to check the sequence. We do it in the bottom-up mode, so it is very well suited. Amino acid composition is normally done by acid hydrolysis and HPLC analysis afterwards, but of course you can couple the LC also to an MS, and the MS proves the mass of the amino acids, so it's not that bad to use also LC-MS here, but the main technique is HPLC. Terminal amino acid sequence, we also, if we have a good sequence coverage, we should get it in the bottom-up mode of LC-MS. Peptide map anyway. Peptide map is peptide map, so this is done by LC-MS. We can, of course, very exactly analyze sulfhydryl groups and disulfide bridges in the bottom-up and middle-down mode when we compare the spectra of reduced sample to non-reduced sample, because in the non-reduced sample we still have the bridges, so peptides are bridged. And then if we reduce, we lose these bridges, so we can directly see which peptides are connected with each other and get a map of the disulfide bridges by this. And of course, carbohydrate structures, I already said that we tend to do this now in middle down mode, but also in the top down mode. If we have good software, we can do the analysis of the carbohydrate structure of our protein. And then, besides the structural characterization, we also have to check for the physiochemical properties. This means that we have to determine the exact molecular weight of our protein. We should look for isoform pattern, extinction coefficient, electrophoretic patterns, LC patterns, and spectroscopic profiles. Okay, molecular weight, of course, top-down mode. We don't digest the protein and we get exact molecular weight if we do it correctly. Isoform pattern are generally analyzed by HPLC and also CE. But of course, both these techniques are very efficiently coupled to MS, so it's possible. And then MS again gives you additional information about your isoform, so it is also a good idea to do this coupled to MS. extinction coefficient is nothing that we do with MS. It is done by the spectrometer, of course. Electric phoretic patterns, I would say STS page and CE is the technique of choice here. Okay, LC patterns, of course, in the top-down mode, for example, we get, of course, the LC pattern in an LC-MS setting. Yeah? And spectroscopic profiles, again, is something that we address with spectrometers. Okay, so, okay, beside all this identity stuff, we said already that we also have to address impurities, and there we distinguish between process-related and product-related impurities. So process-related impurities are, for example, host cell proteins. So if you produce your substance, your protein in a cell culture, you have reminders of so leftovers of the host cell proteins. There can also be leftovers of DNA, for example, from the host cells. Then you have cell culture derived impurities. They come from serum. If you need serum for your culture, then you can have leftovers of serum components. And in some cases, the expression of the protein in the host cell is induced by antibiotics. So you apply antibiotics to your culture, and then the protein is expressed. And then this inducer antibiotics you can also find in your product, and you have to check for it. And then you have, of course, or you can have downstream derived impurities. For example, if you produce your protein as a fusion protein, then you later on want to cleave it with an enzyme. And of course, you can also find, or maybe you find leftovers of the enzyme in your product. Processing reagents that can be reducing agents, for example, and leachables are possible. And yeah, LC-MS is of course perfectly suited for analyzing for cell substrate and also cell culture derived impurities, because if you make a peptide map, you also get the peptides of the host cell proteins, and then you can compare it to a database. And you not only see that there are impurities, but you can also identify the impurities by this technique, what is of course a very big advantage. And also downstream derived impurities, like the enzymes, of course, but also leachables, which we already check for a long time with LC-MS, are seen in the bottom-up mode. So LC-MS is perfectly suited to check for impurities or check the purity of your product. And I said it already, we have process-related and product-related impurities. Product-related impurities are mainly degradation products. This could be truncated forms, for example, because you have hydrolytic enzyme activity in your sample that comes from your host cells, for example, then you have truncated forms and or other modified forms. Oxidation can take place, isomerization, deamidation, or other conjugated forms that you have to check for. And of course, aggregates are possible because proteins, as I said before, tend to aggregate. Truncated forms, top down, middle down, perfect technique for checking for truncated forms. You can identify them, you can identify the mass of the truncated form so that you can, even if you are lucky, say where truncation has taken place and maybe which kind of enzyme activity is responsible for this. Other modified forms also, of course, in the bottom-up mode, perfectly addressed by LC-MS. And aggregates are generally identified by size exclusion chromatography or also CE, capillary electrophoresis. But also in the top-down mode, you can get, it depends really on your product, but in the top-down mode, you can also get an idea about aggregation of your product. And yes, see whether it's dimers or tetramers. Yeah, so it is maybe not that technique of choice, but it can also give you additional information. Okay, so we have, of course, these critical quality attributes that we have to test, and you can make a list that is much longer as the list that I showed you before. I made one. Also, this list is not complete, but it gives a good overview what you might have to do with your antibody or protein product, at least in the beginning, what you have to check. And if you plot this against the techniques that are commonly used in biopharmaceutical analysis, then like LCMS or size exclusion, chromatography, capillary electrophoresis, or ELISAs, then you see that LCMS is really the technique that covers the broadest range of these critical quality attributes. Yeah, so it is really the technique of choice. And there, of course, are other techniques that are specialists for special questions, and it is good to have them in the lab. But LCMS is nowadays really the technique of choice in biopharmaceutical analysis. So we can say that it is a multi-attribute method in biopharmaceutical analysis, and this is why, of course, companies like HWI rely on this technique. Okay, thank you for your attention.